The following is a project supported through the Canadian Agri-Safety Applied Science Program, administered by Agrivita Canada. Developing strategies to minimize health risks in next-generation livestock buildings, integrating modern animal welfare considerations. The increasing public concern for animals to be raised humanely is pushing the livestock industry towards changes in production facilities that improve animal welfare. Changes include adding bedding for animal comfort, providing larger pens where animals are free to roam, and including amenities to allow animals to express more of their natural behaviors. While these changes are positive for the quality of life of animals, they can cause higher levels of gases, dust, and pathogens in the air due to increased animal activity and the presence of bedding materials. These airborne contaminants affect the air quality inside barns and can impact the health and safety of both workers and animals. The main objective of this project is to determine how these new welfare-enhanced production systems affect the air quality in Canadian livestock buildings. Specifically, 3 types of buildings will be evaluated: alternative laying hen facilities, freestall dairy barns, and group housing for sows. Following the air quality evaluation, best strategies for improving the air quality will be selected and tested in commercial livestock facilities to help reduce health risks for animals and workers. For laying hens, there are 2 main types of alternative housing: enriched cages and aviaries. Enriched cages provide more space per hen and include a curtained-off nesting area as well as roosts and scratch pads. These additions allow hens to express natural behaviors such as perching and dust baths. Aviaries are a variation of free-run production systems that provide multiple levels of perching areas in combination with slat flooring. They allow more space per hen and provide an environment where the birds can perch and roost as well as spread their wings. With financial support from the IRSST, the research team sampled buildings using both types of alternative housing as well as barns with conventional battery cages. High concentrations of ammonia, dust of all size fractions, and airborne endotoxins were found in aviaries compared to barns with conventional and enriched cages. The presence of several pathogens and other specific bacteria was tested in the air samples, but differences were only observed for Clostridium perfringens, which was more prevalent in both alternative housing systems. Concentrations of mold, total bacteria, and greenhouse gases were also evaluated, but the results were not significantly different between the 3 types of hen houses. These findings suggest that cage-free houses, such as aviaries, have higher levels of several airborne contaminants that may be hazardous to respiratory health. In order to reduce the airborne contaminants in aviaries, the research team is currently developing a new water-oil acid emulsion to spray on litter and manure. When combined with an absorbent or a heated floor, it should be possible to simultaneously decrease ammonia, dust, and bioaerosol concentrations. In the dairy industry, freestall barn configurations allow cows to move around freely and typically provide different areas for exercise, lying down, and feeding. These systems can also improve udder health, reduce diseases, and sometimes even increase milk production. In conventional tie stall barns, each cow has her own stall, with free access to food and water in an individual manger. Bedding can be added to both housing systems for animal comfort. In-depth air sampling and analysis were carried out in 5 tie stall barns and 5 freestall barns during this project. However, statistical analysis of dust and bioaerosol concentrations indicated no significant differences between the 2 types of buildings. Pathogens and other specific bacteria were analyzed in the bioaerosol samples using qPCR. Airborne E. coli, Enterococcus, Staphylococcus aureus, and many more were detected in high concentrations, but no significant statistical differences were found. Greenhouse gases were similar for both production systems, but ammonia concentrations were highly variable across the different barns. The research team is carrying out a multivariable analysis to evaluate the effect of the production system, barn characteristics, and environmental conditions on gas concentrations and emissions. The results for dairy farms show that the increased movement of cows in freestall barns did not seem to significantly impact the air quality inside buildings. The lack of statistical differences between housing systems could be due in part to the important variations in management and ventilation systems within the same type of barn. The lower animal densities and larger buildings characterizing freestall barns might also have affected results. For pig production, group housing for sows has been suggested to allow animals to express more of their normal activities and behaviors. However, compared to housing sows in individual crates, sows in group housing have more nose-to-nose contact as well as more contact with feces and urine. Sampling has begun in both types of sow housing. Initial results seem to indicate that total dust concentrations are higher in group sow housing. For ammonia, values were actually higher when sows were housed in crates. However, these results are preliminary. Final conclusions will be available when sampling has been completed. The research team is also in the process of identifying and testing reduction strategies for both dairy barns and sow group housing. By comparing conventional and alternative systems, the results from this project will help define reference values and support decision-making during the transition to animal welfare-friendly production systems. Once all the tests are completed, it will also be possible to recommend reduction strategies for commercial-scale operations using alternative housing systems. Please visit our website for more information on all the projects of the Canadian Agri-Safety Applied Science Program.